So welcome everybody. My name is Nola D'Souza. I'm the coordinator for the Australian Fungicide Resistance Extension Network. We're a nationwide network of plant pathologists, fungicide resistance experts and communications and extension specialists. And we're here to provide information to help growers and advisors stay informed about the prevalence and management of fungicide resistance in their regions. We have lots of people this morning. So obviously, you've all come here to see our three experts that we have today. They're here to give you an update of the disease and fungicide resistance in the northern region. But before I start, I just want to do a little bit of housekeeping. There's a few people in the chat today. So what we will do is if you have a question, I'll direct you to your Q&A button at the bottom of your screen. If you could type a question in there, and then I'll put that towards our expert pathologists that we have here today at the end of their presentations. So we would also like and really appreciate the end of our webinar today, we have a survey with a few questions that help us to understand how we have performed, not just in this webinar, but in your general knowledge that you have. This is just so that we can make sure that the information that we're providing is interesting and supporting all of your decision-making processes. It'll take less than 10 minutes of your time, but the feedback that we get is really valuable for tailoring our extension to you. So we'd really appreciate it if you could do that survey at the end. So to start today, based in Wagga Wagga, we have Brad Baxter. He's a research officer in plant pathology for the New South Wales Department of Primary Industries and Regional Development. His work currently focuses on resistance screening of cereal cultivars, and he leads the southern node of disease surveillance and related diagnostics for the Australian grains industry in New South Wales. So I'll leave it over to you. Thanks, Brad.
Thank you, Nola. Good afternoon, everyone. Thank you very much for joining. So this afternoon, I'm going to start off, and we're just going to go through basically what's happening in New South Wales from environmental conditions, what we've seen in regards to our diagnostics and management inquiries that we've had, and also go through some updates regarding some fungicide resistance work that has been undertaken in collaboration with CCDM over the last few years. So hopefully, yes, there we go. So just, I'm not going to read these names out, but just acknowledge that this presentation and the data held within it is a combined effort from these cooperators. So disease triangle, everyone's heard this before. We need a host, we need the environment, and we need the pathogen. So the 2025 wheat crop or cereal crop is going to be our, I guess, susceptible host. The pathogens are present, but rainfall or the environment is the overriding factor. So where do we really sit in New South Wales for 2025? Realistically, we have and have-nots. So when we have a bit of a look, we've got northern and central New South Wales. We've had reasonable starts to the season, whereas southern New South Wales had a very late start. We didn't receive our break until around the third week of May. But since then, we've played a bit of catch up, which has been great. We've received around our average rainfall. So the summer fallow, so we can see the rainfall from 1 November to 30 April in 2024 versus the same time period in 2025. We can see that the summer fallow rainfall has been significantly reduced when we look at 2024 versus 2025. So really, we had, at least in the south, we had a far more average summer, which significantly reduces the risk of volunteers and carryover for our rusts. But where we really start to show up as having a sort of the have and have-nots is when we talk about our post-sowing or our sowing season rainfall from 1 April to 30th of June when we compare 2024 and 2025. With the exception of the very southeast in 2024, we had reasonable and above average rainfall in that period. But when we have a look at 2025, we have really reduced to average or below average rainfall in that time period. So what does that mean? We are entering 2025 or currently existing in 2025 with a lower disease pressure than previous years. But what have we been seeing? So this here is a table of our diagnostics that we've received so far this year. So 2025, so far we've received 192. Just to give you some indication, we did this seasonal update at the start of September last year, and we were closer to 600. So we're far reduced on our inquiries and our samples, given a reflection in the reduction of our disease pressure. So as of the 28th of July, these were our top 10 inquiries and or diagnostics. So of the top 10, we're taking away general disease discussion. So of the top 9, 3 are actually diseases. The other 6 are actually non-disease. So the majority of what we've been seeing so far this year is actually environmental, nutritional, or physiological, with insects and herbicides as well. A lot of the general discussions to this date have been around stripe rust. And you'll notice that when I did this table up on the 28th of July, that we had not received any stripe rust samples. However, that has changed. As of last week, we now have confirmed stripe rust at Armatry, Griffith, Jerilderie, and Lake Cargelligo. And I would suspect that there is other instances of stripe rust out there between those and around those areas that we just are not quite sure of or haven't heard about. So that is sort of the seasonal update. We're existing through 2025. We're coming in at a lower disease level. We haven't seen a lot of disease so far to this point in time, but we are starting to ramp up with our stripe rust and our septoria. So the last part of this presentation is going to be about updating about some of the fungicide resistance work that's been undertaken over the last few years. So you're going to hear these terminologies referred to relatively frequent over the next set of slides. So just as a way of introduction, so we can see over here in a visual that we've got a plate here. So sensitive, the fungicide controls the fungi at label rates. Pretty clean cut. Reduced sensitivity is where it works, but doesn't work optimally. Okay, so we can see that here in the middle where it's restricted some growth, but it's still growing. And in the field, this may be, well, geez, that didn't work as well as last time, or I had to use the higher rate this year to get the same job. Resistant, this one's pretty straightforward. It just does not control the pathogen. Now, lab detection is something that you won't see in the field, but using different techniques in the lab can pick up very sensitive shifts. So this is the Australian fungicide resistance map from 2023. So we can see that this is New South Wales here. We have a little bit going on in barley powdery mildew, wheat powdery mildew, and canola blackleg. But this is completely outdated. And as we can see from some of the other states, we suspected that there was some issues going on with our net blotches that just hadn't been quantified. So we set about filling some of these knowledge gaps of the cereal pathogens in New South Wales. So between 2022 and 2024, we sent away loose smut, net form of net blotch, powdery mildew in wheat, spot form of net blotch, and septoria triticide blotch in wheat. So these have been taken as a part of our crop surveys or diagnostics. And we're under our surveys, we greatly acknowledge the support of New South Wales advisors and growers in letting us get these samples so we can try and fill those gaps. So what have we found? Essentially, so this is a summary of the New South Wales cereal pathogens based off not just our testing, but other testing that's been undertaken by other research groups. But firstly, barley loose smut. We've had some issues with barley loose smut over the last few years. So we sent over 2 years about 32 samples away, and we'll go through those a little bit later, but there was no detections of any reduced sensitivity or resistance to any of the 3 groups there. We did, however, pick up some resistances to group 3s or triazoles, the DMIs, and the group 7 SCHIs in spot form of net blotch and net form of net blotch. And importantly, Septoria triticide blotch, we did not pick up the group 11 strobilurin resistance to date. So this is the updated 2025 Australian fungicide resistance map. So we can see most states have excelled and rocketed through with development of resistances. So we really have to have a good think about what and why and how we're going to combat this to protect the modes of action that we have available to us. So firstly, I'm not going to touch on this too much because Lisle will do a part on IDM, but the next set of slides, I'm going to go through some different diseases and really focus on the mutations and how that affects fungicide choice. But that is the last thing that we should be thinking about when we're coming up with a management plan. So for every pathogen and disease that we're going to talk about moving forward, we always need to start at variety selection in the first place because this is foundational. We then need to bring in other non-chemical farm management, such as rotation, stubble management, management of green bridge. And then lastly, what we'll talk about from now on is the fungicides, but we always need to rotate, use mixtures, and stay within label rates. And really importantly, if we're in doubt, always get a correct diagnosis first. From our diagnostics over the last 5 years or so, each year it's around that 20% are submitted as disease and are actually not disease at all. So it's really important and help is available with that. So first, Septoria triticide blotch. As I alluded to before, there has been no detection of the G143A mutation, which confers resistance to strobilurin. So that's great news. So what we're saying is, until otherwise known, group 11, so strobilurins, are very effective on STB. However, there was a new detection of this particular mutation in Victoria during 2024, meaning that South Australia, Tasmania and Victoria all now have this mutation present. So for us here in New South Wales, it's more likely a matter of when and not if. But until we know, they are still very effective. What we also detected was widespread detection of the CYP51G1 mutation, which affects triazoles. This particular mutation affects all triazoles to differing levels. So I'm going to present a series of tables moving forward. They're all going to be in the same set out where we have year, state, the number of samples, and then the frequency of, so in this case, this is the strobilurin mutation where we didn't detect anything. But then over here, we have the CYP51G1 mutation. And of the 35 samples sent away in 2022, 33 returned this. There was no actual frequency information on those ones. But during 2023, 16 of the 22 samples sent away detected the mutation at an average or mean percentage of 57% and a range of 17 to 94%. Again, in during 2024, 36 of the 36 sent away detected that mutation at an average of 87%. So, but what does this mean effectively? You know, why do we need to know this information? It really is, it dictates how and what we choose to do with our fungicides. So here is just a map here of the frequency of the triazole mutation, CYP51G1. Basically, what we can see from this map is there is no clear pattern to where or how much of that frequency is present. Generally speaking, we would suggest that we would see an increasing level from north going to south and west going to east as the conducive environments do. But this map shows that wherever we're finding some level of septoria, we are still finding high levels of this mutation. So basically what that means is that when we're targeting septoria specifically, we need to be picking robust group 3 actives or triazole actives. Epoxiconazole and prothioconazole are still effective at field rates, but as these are least affected by this particular mutation. However, flutriafol, tebuconazole, propiconazole are all compromised, and ciproconazole is very likely. So net blotches. So here we've got spot form of net blotch and net form of net blotch. Most people would have seen these before. We didn't really understand what was going on in our populations until a few years ago. So we collected samples and sent them away to CCDM. Unfortunately, a lot of the 2023 samples could not be tested, but of the 2 that could, we detected that there was group 3 resistance, triazole resistance, and reduced sensitivity to group 7 SDHIs from these locations. The 2024 testing showed that there was widespread geographical of the mutations relating to both DMI and SDHI insensitivities or resistances. So essentially, we know we have it the length and breadth of New South Wales cropping region. Now, I'm not going to dwell on this too much because I think there was a webinar focusing on this recently, but there is a new triple mutant in net form with insensitivities or resistances to group 3s, group 7s, and group 11s, which has been associated with field failure. Unfortunately, we do not know New South Wales' status at this point in time, but it is something that we're going to try and rectify this year. So again, here we have the frequency information. Frequency information was not undertaken in 2023. It was in 2024. So you'll notice that there is 2 numbers here, one outside the brackets and one inside brackets. So there is some threshold work being undertaken by CCDM to quantify at what level could be classed as a false positive. So in this case, I've just applied a 1%. So anything of a mutational frequency of less than 1% is inside the brackets, and anything outside it is just a pure return of that frequency. So anything from less than 1% up. So either way, whichever way we apply, we are still seeing around 1/3 of the samples had the SDHI mutation in subunit C at around anywhere from 40 to 70% mean frequency. Now, the frequency of the F489L mutation, which affects triazoles, was found in 35 of the 41 samples sent away at around the mean frequency of 27%. At these levels, we would expect to be seeing some level of reduction of efficacy of fungicides. So what can we do moving forward? So we need to avoid putting compromised actives or modes of action into environments where they may select. So we need to further select for that particular mutation. So we need to avoid growing barley in tight rotations and barley on barley. So some of the work that has been done shows that group 3 efficacy, prothioconazole is expected to be the most robust. So that means is expected to still perform at a high, in the presence of a higher level of that mutational frequency than epoxiconazole, propiconazole, and tebuconazole, which is not actually registered for net blotch control. Group 7's bixafen is expected to be better than fluxiproxad, and group 11 efficacy, pyraclostrobin, better than azoxystrobin, but this is based off European work, and from what I understand, some validation of that is going on currently. Just a note that net blotches are seed-borne, so it is possible to transport these mutations and the associated fungicide issues through seed sharing. So lastly, loose smut. I alluded to it before that we did not find anything. So what that means is that a lot of the issues that we had been seeing with loose smut in crop came down to the fact that it was actually a higher level of infection than first thought, no seed treatment, or poor coverage, because there was no evidence of any reduced sensitivity to the indicated actives, which were tebuconazole for the group 3s, fluxiproxad in group 7, or azoxystrobin in the group 11s. So what do we need to do from this point in the season? So a lot of our management practices are set for 2025. Really, we need to look to review 2026, particularly varietal choice. Get out and monitor our crops. In the south, we have a lot of split germinations, which is creating some difficulty in fungicide and herbicide timings. When we're looking at issues in the paddock, we need to look for reasons why it's not disease first, instead of jumping to disease in the first instance. So look how it presents in the paddock. Is it patchy, widespread? Does it follow cedar or spray lines? Appeared suddenly? And have we had the weather conditions for disease to actually develop? A key or a trick is to understand some of the basic biology of common diseases. A latency period is time of infection to expression. This helps to tell us whether it's disease or not, because if we're seeing symptoms of something that is in the paddock that has a latency period like septoria of 4 weeks, and it's on the latest leaf emerged, biologically that can't happen. So it gives us little clues. Usually timing of fungicide is more important than product choice, but product choice is becoming more important for diseases like septoria. Target the key growth stages. Please send rust samples to the Australian Cereal Survey, and let's play the season out. Now, lastly, if you need any assistance, so we have myself, Steve, Zarisa, Job, and Kurt can answer any questions that you need. Now, lastly, thank you for the opportunity to present. The co-authors and co-presenters, and Steve as the project lead, GRDC for investments in New South Wales, Deep Herd for investments under these codes, tech staff, and lastly, growers and agronomists who really are critical to facilitate the support and research by allowing us to access paddocks. So thank you very much. I'll pass over to Lisle.
I'll take over first, Brad. Thank you. That was really great information, Brad, and a lot of things for people to think about. Just a reminder, if you've got any questions, please put them in the Q&A and I can ask Brad those at the end of the webinar if you need to. I'd now like to introduce to you Lisle Snyman. She's a principal plant pathologist with the Department of Primary Industries in Queensland. She's been with the department for 10 years, based in Warwick, where she leads the winter cereal pathology program. Their research and diagnostic service activities mostly focus on foliar diseases of barley and wheat. Over to you, Lisle.
Oh, thanks, Nala. So thank you everyone for joining us today. And yeah, please feel free to ask questions in the Q&A. So I'll talk about barley diseases and then also some IDM practices for disease management. Brad has also touched on this, that we know that environment plays a big role in disease. It will determine, what diseases we get, when will we start to see them, and then also how bad they will be. So if you look at this slide, I've done, just around Warwick because that's where most of our activities are happening. You will see that for 2025, if you compare that to the other years, that we had rainfall higher than the long-term mean for every month except for February. And that also comes on the back of high rainfall in October, November, and December of 2024. So in lots of areas on the Darling Downs is a lot of crops have gone in quite late due to the regular rain events that we had. And then also we do know that if weather is like this, it creates, very favorable conditions for diseases to develop. So for Queensland, what are the key barley diseases, what we are a bit concerned about? So that will be mostly leaf rust, and then the two net blotches, spot form net blotch and net form net blotch. I'm not going to cover powdery mildew. In Queensland, we generally see powdery mildew is a problem early in the season when it's cool and wet, and then it tapers off the moment it starts getting drier and warmer. They do forecast that we're probably going to have a wet spring, whereas generally in our area, August and September tends to be quite dry. So if those conditions persist, then, powdery mildew may become a bit of a bigger problem. The other reason is there's quite a big focus now on the European varieties like IGT Planet and others, and these varieties have quite high levels of resistance to powdery mildew. So I'm pretty sure most of you would be familiar with leaf rust in barley. It's really, really early and easy to, distinguish from any other diseases. It's the most common barley disease all throughout the country, and it occurs in all of the growing regions. It can, cause really significant yield losses in susceptible varieties, and it does also impact quality. The disease spread by means of airborne spores, so that means that it can spread long distances, and it can also create epidemics quite quickly if conditions are favorable. For leaf rust is the green breach is really important. If you have a green breach over summer, especially if we had like a really wet summer, like we had from 24 going into 25, it can, result in high levels of rust very early in the growing season. We do know that the more susceptible a variety, the more disease it will carry, and that will also then result in higher yield loss. If we grow susceptible varieties over a large area, we know it puts increase on the current resistance genes. You can have new pathotypes emerging that then have virulence for those effective resistance genes and result in their breakdown. For barley leaf rust specifically, there has been resistance to several DMIs identified in all of the growing regions. One that our research focus a lot on are the net blotches. It's very important that you're able to distinguish, based on symptoms between these two diseases, and it's really quite easy. Spot form net blotch, in the beginning of the season, both of them, they look very similar. They all start as just tiny little round brownish spots. But then later in the season, as the symptoms develop, it's quite easy to then distinguish between the two of them. Spot form net blotch, it's a spot. They are dark brown to almost like a black color. They're circular and they have a very distinct yellow chlorotic halo around the spots. Net form net blotch, it looks like little ladders, so they're definitely more like a net-like appearance. Both of these diseases can cause very high yield loss in susceptible variety, and similar to most foliar diseases, impact on quality. Both of these are stubble-borne, And we also do know that netform net blotch is also seed-borne, which makes then the carryover from one season to another quite a high risk and emphasize the importance of seed treatment. So I'll talk a little bit about some of the net blotch projects that we're involved in. One is the monitoring of net blotch populations for new virulences of industry significance. This project is led by University of Adelaide and SARDI. So the aim of this project is to equip growers and breeders and basically all of industry to respond to new virulent net blotch pathotypes that are emerging nationally. So as part of this project, there is pathotyping done for both of the net blotch pathogens. So spot form net blotch is done by Agriculture Victoria, and us here at DPI Queensland are responsible for the netform net blotch pathotyping. We receive samples from collaborators all over the country that include pathologists, agronomists, breeders, and also growers. So you'll see if you look at the bottom there, 2024 has been a really big season for netform net blotch across most of the states, and we received 111 samples in total. We pathotype these isolates on a set of 30 differential genotypes, and then we use the infection responses to determine the specific isolate and the variation between these isolates. So there's three main isolates that are detected across the country. They are NB 85, 73, and 350. And you will see, if you look at the blue one, NB 73, there has definitely been a big increase in this specific isolate over the last few seasons and a decline in the other two. We are still busy because we received such a huge amount of samples for 2024, so we haven't finished the pathotyping of that yet. So we do not have the final and analyzed results for that, but we do expect that that tendency will continue. If you look at the pathotyping results over the 4 years, unfortunately, my year dates have gone walkabout here. So they are top left corner is 2020, bottom 2021, and then 2022 and 2023 in the right bottom corner. So if you look at that, you will see that the blue one, NB73, that's an isolate that was common in Queensland for a very long time. It's the one that we call the old shepherd isolate. But if you look across the years, you will see in 2020, it was kind of really only present in Queensland, but it has spread all across the country, and it is now by far the most dominant isolate that's detected across all of the states. And that is an indication of the cultivation of the European barley varieties, and they are specifically susceptible to this isolate. And as I mentioned before, I think that that is, as long as we have such a huge component of the European varieties in our cultivation, that is something that we will see increase into the future. As you can see, it's very important for us that we keep on doing this work because it gives us an indication of really what is going on out there. So we do ask everyone, if possible, please send us net blotch samples. We will process the netform net blotch ones in Warwick, and the spot form net blotch ones we pass over to Agriculture Victoria. With any samples that you send us, please send us as much information as possible. Whatever you can get your hands on, you'll see there's a huge list there. Variety for us is really important, and then also location. You can send the samples to either Hermitage at Warwick or Lisle in Toowoomba. Please mark them for my attention. You can either mail them, drop them off. And if you do have problems that you can't get samples to us, just give me a call and we will see what we can arrange. If you collect samples, please send them to us as soon as possible. please don't put them on the dash of your ute for days on end. They do crisp up and then we can't really work with them. And also no plastic. If there's any moisture around, they will just go moldy. Remember that we also do offer some diagnostic services. And as Brad has alluded to as well, especially in years like the current one, where we have cold or cool conditions, very wet conditions early in the season, we do see a lot of physiological leaf spotting and also some nutrient deficiencies that could be, just misdiagnosed on farm level as disease. So please contact us. We can have a look at samples. Initially, even if you can just send us some photos, we could have, like an indication of what it can be, but it may be that we need to get our hands on some samples so that we can make a conclusion. Rust samples, yes, please send them away for the cereal rust survey at the Plant Breeding Institute at Sydney Uni. If you do send it on to us, we will just pass it then to them, which delay the whole process. So there's another project also on the net blotches that we're involved in, is integrated management strategies for net blotch in low, medium and high rainfall zones. So we do lead that project. Know that the net blotches, like all other diseases, impact yield and quality and decrease grower profitability, but we don't really know the extent of that, and we don't really have an idea about cost-effective control strategies in the different rainfall regions. So the aim of this project is to develop integrated disease management strategies and also determine the cost benefit of implementing these in each of the rainfall zones, and then also to deliver an extension strategy to drive grower adoption. We do know that fungicides are quite important in terms of controlling disease, but we do need to know when and when not they are cost effective. And then also with all of the changes that are happening, we do need to look at the efficacy and sustaining efficacy of fungicides. So for these trials, we have collaborators in a few or all of the eastern states, and trials are conducted in New South Wales, Queensland, Vic and SA, and it covers all of the different rainfall zones. So we look at different varieties with different net form net blotch ratings. So that would be varieties that are suited to the specific regions. We look at using seed treatment and also net form net blotch in the absence of ineffective seed treatment, also different mode of action. For foliar fungicides, also looking at different mode of action, but then also times of application, because we do know that these are variable across the rainfall regions. There are trials conducted in clean stubble versus infected stubble, and then we also compare that with trials conducted on fallow land. University of Southern Queensland are looking at changes in the pathogen population over time in some of these trials. So I'm not going to dwell on this. Brad has talked about that, and my next slide will discuss some of these principles in a bit more detail. So these are management strategies specific for the net blotches, but it's important to remember that most of these you can apply to all of the foliar diseases of barley. Variety choice is really important. We always emphasize that you try and use a variety with at least an MS rating, and also look at varieties that are suitable to your specific region. The NVT database on the GRDC website have all of the information for that. And then, you plan your management strategies according to that specific variety. We did see with the pathotyping that variety have a big impact in terms of pathotypes that are around, and we also know that's the case with stripe rust in wheat as well. So the other thing that you can look at is crop rotation. That will limit the impact of disease. It's best to rotate with non-host crops such as wheat, chickpea, mung bean, sorghum, or whatever is suitable for your specific area. We do know that stubble plays an important role in terms of the net blotches because both of the diseases are stubble-borne. And remember that if you make the decision to plant barley back into barley stubble, especially if you know that that barley from the previous season had net blotch in it, you do run the risk of having high levels of disease early in the season. If for some reason you really have to do that, it's your best paddock and you want to go back into that barley stubble that you had last year, make sure that you at least try and use a different variety. However, that is something that we would just never recommend. Also remember that the environment plays a big role in the breakdown of stubble, and especially if conditions are dry, then stubble breakdown occurs at a much slower rate. So don't necessarily only look at what you had in the paddock last season, but also at seasons before then. We do know that early season sowing tends to favor disease. I think with a lot of the crops in southeast Queensland going in quite late this year, it still seems to be a bit of a quiet year with diseases only starting to move now. Many pathogens over summer in the green bridge, and if you don't control that, you are likely to have high levels of disease early in the season. And we do know that that green bridge is very important specifically for the rust diseases. Brad has also touched on crop monitoring. Don't just rely on the agronomist. Like every one of us, agronomists are time poor. They don't really have the time to walk up and down through every single paddock and cover your whole paddock. And that's your responsibility as a grower to make sure that you know what's happening in your paddock. And then also you'll be able to provide us with the information if there's issues that you think they may be disease, like where are they? Are they on the low-lying areas? Are they in patches, or is it spread across the paddock? So please make sure that you do spend enough time in your paddocks. Fungicides, that will always be part of our IDM strategy, but we do know that we are running into a lot of areas, running into problems with fungicides that are just not effective anymore. So if you do have to use fungicides, only apply them when needed. So only if you have disease. I know it's a general practice to put a fungicide in with early herbicide applications. And I think in terms of fungicide resistance developing, that's really something we need to reconsider. On the other hand, it's also important not to wait until, your crops are really absolutely flogged with disease. We do know that the fungicides are much more effective at lower disease levels. The other thing is that net blotch is also seed-borne. So an effective seed treatment, we know that that can limit the carryover of disease from one season to the next, and it can also delay that early onset of disease. So these IDM practices, as I said, they are basically, hard and fast rules for all of the foliar diseases, depending on a few factors. So, and remember that fungicides are important for us, and the way that we use them and we manage them will determine their longevity and their effectiveness in future. There aren't new ones coming along every year, so we have to protect what we have. So yeah, thank you so much for taking the time to attend. And like I said, my contact details are there on the screen. Don't hesitate to contact us. Please send us samples and let us know if you have any problems that you would like answered. We also welcome going out to farms and having a look at your crops. So thank you very much. I'll pass on to Nola.
Thanks, Lisle. That was really lots of good information that I'm sure everybody managed to get some really good things out of that. Thank you so much. Now we'll move on to Professor Levente Kiss, the Director of the Centre for Crop Health at the University of Southern Queensland in Toowoomba. As a plant pathologist, Levy's current focus is on obligotrophic plant pathogens, such as powdery mildews, rusts and downy mildews, and mycoparasites. He also has expertise in fungicide resistance, of course, and weed control. Thank you, Levy. I'll put that over to you.
Thank you for the introduction, Nola. Good afternoon, everyone. As the title suggests, I will mostly focus on a single crop, mung bean, and I will talk about results of a GRDC supported project that ended last year. And I will also present the latest results, the most recent results from a number of projects supported by the Broadacre Cropping Initiative, which is a partnership between Queensland Department of Primary Industries, formerly DEF, and our university. So I don't think the major diseases of mung bean need a lot of introduction. There are 2 bacterial diseases, 3 important fungal diseases, and the nasty root lesion nematodes that cause or may cause serious yield losses every year in mung bean paddocks. Amongst these, there's just one disease, the powdery mildew, that requires every year fungicide management. I won't go into detail because the disease is really common and well known, and by the end of the season, sometimes crops look like they are painted in white, like on this image. And this disease can actually cause up to 40 or even 45% yield losses if the epidemic starts before or during flowering, if the environmental conditions are conducive during the whole season, and if the disease is left unmanaged. Now, the disease at the end of the season may also have an impact on harvest management because it may impact the desiccation efficacy. Basically, the desiccant cannot be taken up if there is not enough green tissue there in the paddock. In terms of management, disease management, we have a non-chemical management opportunity if the crop is sown early in summer and goes through the flowering and even the green pod stage before temperatures start dropping at the end of summer and beginning of autumn, then the powdery mildew epidemics won't have an impact on yield. So this is an easy way to avoid powdery mildew if otherwise it is suitable to sow early. The other general management option is, of course, using resistant crop varieties. However, in mung bean, there are no resistant crop varieties currently available. And I would like to just mention Dr. Mary Ryan's team's new varieties, Broga and Kookaburra, that were released earlier this year. These varieties and also the other varieties have a limited genetic resistance to powdery mildew, and all current mung bean varieties still require fungicide treatment. If, as I mentioned earlier, the disease comes during or before flowering. In terms of fungicide management, we have 2 mode of action fungicide registered or available under permit to manage mung bean powdery mildew. One is the good old tebuconazole, DMI or an azole fungicide, mode of action group 3. And we have Veritas Opti, which is a cocktail of TEB and azoxystrobin. And azoxystrobin, as Brad has already mentioned, it's a QOI or group 11 fungicide. So these are the 2 mode of action fungicides that we have available. Now, the evergreen question is when to spray. And of course, people have a lot of experience about this. And we also have an app, freely available app in both Apple Store and Google Play that was developed by the Western Australian government's Department of Primary Industries and Regional Development with GRDC support. So this app, I do recommend it to use it. It actually calculates the net return, if there is any net return from each plant spray. And as I said, it's free, it's easy to use, it's reliable. We have tested it in a number of scenarios, and it is based on the philosophy that if it doesn't pay, don't spray. So spray only for financial benefit. And this also is a good idea also from a fungicide resistance perspective. So we answered the question when to spray. Now comes the question what to spray. And this question was mainly researched in the GRDC supported project that I mentioned earlier. We have set up a number of replicated field trials with 4 replicates per treatment in a number of locations in Toowoomba and around Toowoomba in southern Queensland. These are the treatments here in this table. We had, as I said, 4 plots per treatment. So 4 plots at random design received 1 spray of tebuconazole at first sign of disease. This was treatment number 1. Another 4 plots, 1 spray of Veritas Opti. I said they are low rate, 250 milliliter per hectare, but this is not a low rate actually. It's a middle range rate according to the label. So 1 spray with Veritas, first sign of the disease. Then treatment number 3, 2 sprays of tebuconazole, first time of the disease, and then approximately 2 weeks later, treatment number 4, 2 sprays with Veritas, the same 250 milliliter per hectare treatment. Then we introduced what we called the common industry practice 1, which was 1 spray with tab and insecticide at first sign of the disease, and then a second one approximately 2 weeks later. Treatment number 6 was the control, the so-called untreated control, although this was actually treated with the surfactant only. Then we introduced what we called common industry practice 2, which was 1 preventive spray with tab, then a second one approximately 2 weeks later, and then a third one approximately 2 weeks later, again, all with fungicides. And then treatments number 8 and 9 were 1 or 2 sprays with Veritas Opti at the highest label rate that is recommended for mungbean. So Veritas High 1 and Veritas High 2. And these experiments, these field tests were carried out by this team in alphabetical order. Dr. Kirsty Owen from UniSQ, yours truly, Dr. Lisa Kelly from Queensland Department of Primary Industry, and almost Dr. Neil Robinson also from UniSQ. This is an image put together by Neil of one of the field trials. So you will see here the 4 replicates of each treatment, and the treatments shown in red are, well, I put there no sprays, but these were sprayed with water and surfactant. So those were the non-treated controls. And the other end of the spectrum are those plots that were sprayed with Veritas High 2 times. And already the color of the canopy indicates a huge difference. Obviously, the control plants were almost completely painted in white, while there was much more green canopy in those that were sprayed with Veritas High 2 times. And this will be the only graph that I'm going to show you today, which is a simple way of showing the grain yield response to different fungicide treatments compared to the untreated control. And bars shown with asterisks are those that indicate that the yield was significantly greater than the untreated control. So first of all, it's really good to see that in this particular experiment, up to 44% grain yield loss was actually protected with the fungicide treatment. And that was Veritas applied at the highest possible labor rate 2 times. The other thing that is also important here is that only these treatments, these 3 treatments, resulted in yield responses that were significantly higher than the untreated control. And we can have another look at this graph. And we may see that actually we are sitting on the edge, because tebuconazole only once, twice, and even 3 times. Yes, there was a grain yield response to tebuconazole, but that was not significantly higher than the untreated control. So the fungicide did its job, but certainly not as expected, because we would have expected, obviously, a significantly higher yield across the experiments in southern Queensland. So this was consistent along these experiments. So we wanted to know more about these field failures of tebuconazole, and I talked a lot to Fran Lopez-Ruiz, the leader of AFREN at CCDM, and we had to agree that yes, these should be treated as field failures. Let's see if there are mutations in the powdery mildew populations that actually support what we found in these replicated field trials. So we did 2 things. We have collected powdery mildew samples with these punctures, as you can see, leaf discs covered with powdery mildew at random in mung bean paddocks. And then we extracted DNA from these samples and looked for known mutations in the powdery mildew populations. We have also looked for powdery mildew colonies that survived even sprays 2 times with the highest labor rate of Veritas. And we found such colonies, and then from these colonies, we isolated single powdery mildew spores, and from these single spores that germinated, as you can see on this image, we produced single spore isolates and maintained them in isolation. Then with the support of the Broadacre Cropping Initiative projects, we looked at single point mutations in all these mung bean powdery mildew samples. And we confirmed that mutations associated with the QY resistance and mutations associated with the DMI reduced sensitivity were found in populations of one of the causal agents of this disease, Podosfera xanthii. You may remember at the beginning I mentioned that 2 powdery mildew species cause this disease. And we found the QY resistance associated marker also in the other species. We still don't know if the DMI reduced sensitivity associated mutation is present in the other species. And a large part of this work was done by our wonderful PhD student, Ayesha Senanayake. You can see her here collecting mung bean samples for fungicide resistance tests. Now, this is not the only thing that we have done. Ayesha and the team has started to develop methods to quantify or to determine the frequency of the mutations in mung bean powdery mildew populations across southern Queensland. And you can see here the first data. The wild type is the powdery mildew that doesn't have a mutation associated with DMI reduced sensitivity, while the mutant is the one that has 1-point mutation. And we still don't understand fully the mechanisms, the molecular background of the DMI reduced sensitivity in mung bean powdery mildew. But in terms of the frequency of the mutations, now we have some data, and the frequency of these mutants is quite alarming, as you can see on these graphs. If I move to the next slide, yes. Oops. Too much. Yes, we have also done classical fungicide resistance tests in the laboratory with these isolates that were produced from single spores after being sprayed with fungicide. And I don't think I can move my mouth on the screen, but you can see on the right-hand side, top right-hand side, there are mung bean leaves, the first true leaves of the mung bean, treated with 1/4 of field rate of tebuconazole, half field rate, full field rate, and 2 times field rate of tebuconazole. C is the control, and you may see powdery mildew colonies developed after 2 weeks after inoculation on the control in the wild type, but the powdery mildew without the mutation was not able to grow even on a 1/4 field rate of tebuconazole. However, the other line shows a monosporic or a single spore with the mutation, and the colonies are the same size on the controls, and then there are small powdery mildew colonies even on leaves sprayed with 2 times the field rate of tebuconazole. Those are circled with red on those leaves. So this is not fungicide resistance, but this is certainly reduced sensitivity, as Brad introduced it in his presentation earlier. So the fungicide works, but certainly cannot fully control the pathogen anymore due to this mutation. So again, we don't fully understand the mechanism of this DMI resistance. It could be other things there in the background, not just this mutation, and we are exploring these questions at the moment. But coming back to our problem, why to worry? Fungicides work after all. If we look again at the same graph, we can see there nice yield response to the fungicide applications, although we are sitting on the edge. However, I think that time to act is now, when the fungicides are still working, but certainly the mutations are there and the pathogen populations are evolving. What do I mean by act now? Both Brad and Lisle introduced the fungicide resistance 5. So I will just go through these 5 recommendations from the point of view of mung bean powdery mildew. So the first recommendation is avoid susceptible crop varieties. And in cereals, there are many more options. In mung bean, basically all currently available varieties are susceptible to the disease to some extent. Rotate crops and reduce the disease carryover. Well, we are dealing with a fully airborne disease here, so rotation won't really help in this particular case. Use non-chemical control methods. Yes, there is one, and this is plant early in summer if at all possible. Then spray only if necessary and apply strategically. There is the app that will help this strategy. And the last recommendation is rotate and mix fungicides. Well, there are only 2 mode of action groups, and there are only 2 fungicides, azoxystrobin and tebuconazole, that are both quite old products on the market. So I don't want to sound dramatic, or maybe I want to sound dramatic, because I think that we are soon running out of options in terms of mung bean powdery mildew management, fungicide management. When I say soon, I don't think a year or 2, but certainly there are mutations. Certainly we have documented field failures, at least with tebuconazole. So probably there is time to look for fungicide products that have other mode of action actives, and just test them if the industry thinks that this is needed. There are just 2 reasons to look for new actives. One is just to get ready with alternatives if TEB and azoxy fail due to fungicide resistance. And second, let's just simply compare the efficacy of the current options to new actives, because maybe there are better methods of fungicide management of mung bean powdery mildew. And now this will be indeed dramatic. I think that without perfectly working fungicides, powdery mildew in itself, because it may cause up to 40% yield loss, right? So in itself may strongly reduce the profit margins of mung bean production. So fungicide resistance in this particular disease, I think it should be taken very seriously. Is there anything else we can do? So I would like to just raise one more thing, because I'm not sure about the appetite for something that can be called a biofungicide. So what is a biofungicide? Obviously, not a chemical fungicide, a fungicide that is based on a living organism that controls the pathogens. And powdery mildews have their well-known and very well-researched natural enemies. These are fungal hyperparasites. And overseas strains have already been developed into commercial biofungicides in the European Union, the USA, Korea, India. There are different products based on different strains of these fungal hyperparasites. Here is just one product that is used and applied in mostly against grape powdery mildew in the European Union, the EQ10 biofungicide. So these are all overseas products and based on overseas strains. However, we found these hyperparasites in mung bean powdery mildew samples and also in other powdery mildews here around Toowoomba in southern Queensland. So they are there and they are suppressing powdery mildew infections to some extent. A selected strain of these hyperparasites may become Australia's very first locally developed biofungicide product. Some uplifting music here, please. If, of course, there is industry support for such an approach. So there is another way of probably of controlling this disease with a non-chemical fungicide, a biofungicide. We are running out of time, so I won't read all these conclusions after this presentation, because these would just simply repeat what I have already told you. How about this wrap up? You may know this famous T-shirt that is available somewhere. It says, "What doesn't kill you mutates and tries again." And I think if we change it a little bit, what doesn't kill your crops that were sprayed will mutate and will try again, and then it will infect your crop again. So I think this is a good summary of what we were talking about here today during this hour. And my very last slide is just further reading. This year, a fact sheet was developed and also a paddock practice that are available and have more information about mung bean powdery mildew management. Thank you very much.
Thank you, Levy. That was a lot of really good information, and I'm sure that quite a few people will be thinking about that, especially that good advice of not putting our heads in the sand about fungicide resistance and taking advantage of the IDM strategies that you presented here and all of our pathology experts presented here today. So I'm aware of the time, and there are no Q&A questions at the bottom there. So what I might do is wrap this up now. I'd like to thank, on behalf of all of you, Levy and Lisle and Brad for giving their insights into fungicide resistance management and an update in the northern areas. I'd also like to thank all of you for staying on with us this long, and we'd really appreciate it if you could finish with our survey at the end of this if you have the time, because that would be really great feedback for us to make sure that what we're presenting to you is exactly what you need. So thank you, everybody, and we will see you in our next webinar. Thank you.